Ladies and gentlemen, good evening. Welcome to the 17th meeting of the Brazilian Society of Toxinology in conjunction this year with the FPTX. And finally, Professor Mariana Castro, President of the SPTX. Thank you, welcome. And I will now give the floor to Professor Mariana Castro.
Boa noite a todos. Em primeiro lugar, eu queria dar as boas-vindas. Espero que vocês aproveitem bastante essa oportunidade e que ela traga, né, frutos para a maioria de vocês, que eu acredito que tenha vindo aqui para obter, né, uma fonte de conhecimento e de oportunidades de intercâmbio. E, nesse momento, eu gostaria de dividir a palavra com os meus colegas, principalmente com o professor Julian White, que é o presidente da nossa principal sociedade em toxinologia, a IST, e ao término, né, quando a gente tiver já um pouquinho mais adiantado nessa sessão de abertura, eu vou fazer um discurso um pouquinho mais aprofundado. So, let's change to English. Sorry about my rusty English. Because of the pandemics, I was away from English for several years. I was trying to learn Italian, and I enjoy it a lot. But okay, I would like to say hello to all of you and be welcome to our meeting. I hope this opportunity brings to all of you a source of knowledge and also opportunity to establish new partnerships. This initial talk, I would like only to open the ceremony. I will share this talk with Professor Julian White. He is the president of our main society, the International Society on Toxinology, and I would like to share this initial talk with him. And after these preliminary considerations, I will bring to you my full speech. So, at this moment, I would like to pass the honor of the speech to Professor Julian White.
Thank you. Firstly, I wish to welcome you all here to this 13th Congress of the Pan-American section of the International Society on Toxinology and the 17th Congress, 17, wow, 17th Congress of the Brazilian Society on Toxinology. This is the 4th time I've been to Brazil for a Congress. 3 of those times, including this time, are to do directly with toxinology, the IST, and the Brazilian Society. And they've all been very interesting congresses. This Congress has occurred after a difficult period for the IST and many organizations internationally as a result of the pandemic and now tragically an epidemic of wars that has disrupted our planning and that of so many people around the world. Certainly we felt from outside of Brazil that there were some difficulties with the gestation of this particular Congress. And we were worried up until very recently about what the birth would be. But it seems, looking at all of these faces here today, we shouldn't have worried so much. And so the first thank you I wish to give is to all of you, because without you, the people attending this Congress, we wouldn't have a Congress. I'd also like to thank the various speakers from Brazil and from other parts of the world, and especially some of the senior members of the IST who are either here already tonight or will arrive later in the week. And I have to say, we have not one but two ready awardees attending this Congress. They are here with us tonight, Juan Calvet from Spain and Bruno Lamonte from Costa Rica. So to attract speakers of this caliber and this reputation does credit, I think, to the organizers of this Congress. So I therefore wish to thank Mariana and all of her colleagues who have put, I'm sure, so much hard work into making this Congress happen, to making sure that we are here today, either in person or virtually. And you can see the equipment here. I think it's very important in the future that IST Congresses have a virtual possibility, a hybrid Congress for the people in other parts of the world that can't easily attend. I'd also like to thank the members of IST, particularly on IST Council, who have worked with the Brazilians and with myself as president, supported me as president in making this Congress and the last one, our World Congress in Abu Dhabi last year, a possibility. Rather than bore you with more thanks, etc., I would rather now retire from speaking and look forward to our first presentation. So lastly, once again, thank you, Mariana. Thank you to all of your organizing committee and to all of you here today.
And now I would like to invite Dr. Elda Sanchez to give her welcome words.
I will definitely be brief. I just want to welcome everybody again and thank you very much for attending this important conference. And I hope that we can come together and create friendships and collaborations so that we can continue this wonderful work that we have and that will continue for years on. So thank you very much for attending.
Okay, so now it's up to me. Vidal, would you like to give them? Okay, Vidal is shy. So I will change my position and give you my speech. Okay, so if you prefer, okay, be. I will try to change from Portuguese to English during my speech, okay? First, em português. Primeiro, em português. Senhoras e senhores, estimados cientistas e, principalmente, estudantes que estão entrando em contato com a toxinologia. Eu estou profundamente honrada em estar aqui hoje, diante de vocês, para discutir, inicialmente, a importância de duas figuras que deixaram uma marca indelével na ciência brasileira e internacional. Vital Brasil e Sérgio Ferreira. Esses dois indivíduos notáveis fizeram contribuições significativas ao campo das toxinas animais, um domínio que abriu novos horizontes para a descoberta de medicamentos e enriqueceu enormemente nossa compreensão da extraordinária biodiversidade do Brasil. Ladies and gentlemen, esteemed scientists and mainly students who love toxinology and want to learn more about them. I am deeply honored to stand before you today to discuss the immense importance of two figures who have left an indelible mark on Brazilian and international science, Vital Brasil and Sérgio Ferreira. These two remarkable individuals have made significant contributions to the field of animal toxins, a domain that has opened many new horizons for drug discovery and greatly enriched our understanding of the extraordinary biodiversity of Brazil. Vital Brasil, nome sinônimo de exploração da biodiversidade brasileira, foi um cientista visionário que dedicou sua vida ao estudo dos organismos peçonhentos e de suas toxinas. Nasceu em 1865, em Minas Gerais, no Brasil, e é talvez mais conhecido por seu trabalho pioneiro com venenos de serpentes e pelo desenvolvimento dos soros antiofídicos. Seus esforços incansáveis e pesquisas inovadoras salvaram inúmeras vidas, fornecendo meios para tratar envenenamentos por picadas de cobra. O seu legado não se limita à criação dos antivenenos, que salvam vidas, mas estende-se ao campo mais amplo da toxinologia. Seu trabalho lançou as bases para a compreensão das complexidades dos organismos peçonhentos e de suas toxinas. Sua pesquisa foi fundamental para descobrir o potencial terapêutico oculto nessas toxinas. Através de sua dedicação inabalável, ele abriu caminho para que pesquisadores aproveitassem o poder das toxinas para o desenvolvimento de medicamentos e terapias que vão além dos soros, abrangendo diversos campos, como controle de dor, tratamento de câncer, neurologia, Vital Brasil, a name synonymous with the exploration of Brazilian biodiversity, was a visionary scientist who dedicated his life to the study of poisonous organisms and their toxins. Born in 1865 in Minas Gerais, Brazil, he is perhaps best known for his pioneering work with snake venoms and the development of antivenom serums. His tireless efforts and groundbreaking research have saved countless lives by proving the means to treat snake bite envenomations. But his legacy is not limited to the creation of life-saving antivenom, but it extends to the broader field of toxinology. Vital Brasil's work laid the foundation for understanding the complexities of venomous creatures and their toxins. His research has been instrumental in uncovering the therapeutic potential hidden within these toxins. Through his unwavering dedication, he paved the way for research to harness the power of toxins for the development of drugs and therapies that go beyond antivenoms, touching various fields such as pain management, cancer treatment, and neurology. Outra referência no mundo das toxinas animais é o professor Sérgio Ferreira, renomado farmacologista brasileiro, cujas contribuições repercutiram internacionalmente. O trabalho do professor Ferreira concentrou-se nas peçonhas de algumas das criaturas mais fascinantes e mortais encontradas no Brasil, incluindo a peçonha da serpente jararaca. Sua pesquisa pioneira sobre proteínas presentes na peçonha de serpentes e sua interação com o corpo humano foi transformadora. Ele descobriu o peptídeo conhecido como fator potencializador da bradicimina, que revolucionou nossa compreensão de como as toxinas podem ser usadas como ferramentas para a descoberta de medicamentos. Another reference in the world of animal toxins is Professor Sérgio Ferreira, a renowned Brazilian pharmacologist whose contributions have reverberated internationally. Professor Ferreira's work has focused on the venoms of some of the most fascinating and deadly creatures found in Brazil, including the venom of the jararaca snake. His pioneering research on snake venom proteins and their interaction with the human body has been transformative. He discovered the peptide known as bradicinin potentiated factor, which revolutionized our understanding of how toxins can be used as tools for drug discovery. As descobertas inovadoras de Ferreira na década de 60 levaram ao desenvolvimento dos inibidores da ECA, uma classe de medicamentos que revolucionou o tratamento da hipertensão e das doenças cardiovasculares, melhorando a vida de milhões de pessoas em todo o mundo. É importante destacar que essa classe de medicamentos é ainda hoje uma das mais prescritas no mundo. Ferreira's groundbreaking discoveries in the 60s, last century, led to the development of ACE inhibitors, a class of drugs that revolutionized the treatment of hypertension and cardiovascular disease, improving the lives of millions worldwide. It's important to highlight that this class of drugs is, until now, one of the most prescribed in the world. Esses dois pesquisadores, Vital Brasil e Sérgio Ferreira, demonstraram que as toxinas animais, antes vistas como ameaça, também são tesouros de potencial incalculável. O seu trabalho mostra como o estudo de organismos venenosos e peçonhentos pode produzir conhecimentos extraordinários sobre biologia e medicina. Suas conquistas são emblemáticas da rica biodiversidade do Brasil e do papel crítico que o país desempenha na toxinologia. These two luminaries, Vital Brasil and Sérgio Ferreira, have demonstrated that animal toxins, once seen as threats, are also treasures of untold potential. Their work showcases how the study of poison and venomous organisms can provide extraordinary insights into biology and medicine. Their achievements are emblematic of the rich biodiversity of Brazil and the critical role the country plays in toxinology. Além disso, seus trabalhos ressaltam a importância de se preservar os ecossistemas e a biodiversidade únicos do Brasil, já que eles são a fonte de muitos organismos notáveis e de suas toxinas. Nós, como cientistas, somos guardiões desse tesouro de conhecimento e temos a responsabilidade de garantir a sua proteção para gerações futuras. Furthermore, their work underscores the importance of preserving the unique ecosystems and biodiversity of Brazil, as they are the source of many of these remarkable organisms and their toxins. We, as scientists, are custodians of this treasure, trove of knowledge, and have a responsibility to ensure its protection for future generations. Os legados de Vital Brasil e Sérgio Ferreira servem como fonte duradoura de inspiração para todos nós engajados na exploração da biodiversidade brasileira e no estudo de toxinas em geral. As suas contribuições para a ciência salvaram vidas, avançaram a nossa compreensão da biologia e da medicina e remodelaram fundamentalmente o panorama farmacêutico. Estamos sobre os ombros de gigantes e, ao nos reunirmos aqui no Brasil, deveríamos levar adiante a tocha deles, continuando a desvendar os segredos dessas notáveis toxinas para a melhoria da humanidade. As toxinas animais encontradas numa ampla gama de organismos, desde cobras e aranhas até caracóis e sapos, têm cativado o interesse humano durante séculos, devido aos seus efeitos potentes e muitas vezes letais. No entanto, hoje olhamos para além da sua natureza mortal e nos concentramos no potencial que possuem na definição do futuro da medicina. The legacies of Vital Brasil and Sérgio Ferreira serve as an enduring source of inspiration for all of us engaged in the exploration of Brazilian biodiversity, in our case, and the study of toxins in general. Their contributions to science have saved lives, advanced our understanding of biology and medicine, and fundamentally reshaped the pharmaceutical landscape. We stand on the shoulders of giants, and as we gather here in Brazil, we should carry their torch forward by continuing to unlock the secrets of these remarkable toxins for the betterment of humanity. Animal toxins found in a diverse range of organisms, from snakes and spiders to cone snails and frogs have captivated human interest for centuries due to their potent and often lethal effects. However, today, we look beyond their deadly nature and focus on the potential they hold in shaping the future of medicine. Nós também devemos fazer referência às mudanças que enfrentamos durante essa era pós-Covid-19. A pandemia redefiniu o modo como fazemos pesquisa, colaboramos e disseminamos conhecimento. Enfatizou a importância da adaptabilidade e da resiliência na comunidade científica. A nova geração de estudantes que vão crescer nesse novo mundo transformado por essa crise vai ter um papel crucial em definir o futuro da ciência. We must also acknowledge the challenges we face in our post-COVID-19 era. The pandemic has redefined the way we conduct research, collaborate, and disseminate knowledge. It has emphasized the importance of adaptability and resilience in the scientific community. The next generation of students who have grown up in a world transformed by these crises will play a crucial role in shaping the future of science. Nessa nova era, nós devemos encorajar e apoiar as novas mentes que venham a embarcar nessa jornada de seu próprio conhecimento científico. Elas trazem perspectivas novas, habilidades tecnológicas e a capacidade de realizar colaborações de modo interdisciplinar. Nós, como pesquisadores experientes, temos a responsabilidade de orientar, guiar e empoderar esses novos pesquisadores. Suas descobertas e inovações irão, sem dúvida nenhuma, ser a chave para resolver a maioria dos desafios prementes do nosso tempo. In this new era, we must encourage and support these young minds as they embark on their own scientific journey. They bring fresh perspectives, technological expertise, and the capacity to drive interdisciplinary collaboration. As experienced researchers, we have a responsibility to mentor, guide, and empower them. Their innovations and discoveries will undoubtedly hold the key to solving some of the most pressing challenges of our world. Agora que nos movemos adiante nessa nova era pós-Covid-19, eu gostaria de lembrar que as descobertas científicas, elas precisam ser transmitidas para as novas gerações. Juntos, nós podemos desbravar as fronteiras do conhecimento, superar obstáculos e trazer terapias transformadoras para aqueles que as necessitam. Hoje, reunidos aqui em Brasília, nós vamos reconhecer o incrível potencial que as toxinas de origem biológica apresentam e o extraordinário impacto que elas podem ter no futuro, em particular da medicina. Eu peço a todos vocês que continuem suas pesquisas nesse campo excitante, descobrindo esses segredos contidos nessas moléculas e avançando nos limites relacionados com a descoberta de novas drogas e de alternativas terapêuticas. Juntos, nós podemos construir um mundo mais saudável e melhor através do poder das toxinas que nos foram oferecidas pela natureza. Eu gostaria de agradecer a todos vocês pelo esforço de estar aqui hoje e que a gente consiga efetivamente garantir, né, um mundo melhor e mais saudável para todos. E agora eu declaro esse congresso aberto. As we move forward into this new era post-COVID-19, let us remember that the torch of scientific discovery is passed to the new generation. Together, we can continue to push the boundaries of knowledge, overcome obstacles, and bring transformative therapies to those in need. And as we gather here today in Brasilia, let us recognize the incredible potential that biological toxins hold and the extraordinary impact they can have on the future of medicine. I urge all of you, fellow scientists, to continue your research in this exciting field, unlocking the secrets of these fascinating molecules and pushing the boundaries of drug discovery and therapeutic approaches. Together, we can make the world a healthier and better place through the power of nature's toxins. Thank you, especially because of your collective efforts to be here today and to bring a brighter and healthier future for all of us. And now, I declare this meeting open. Thank you. I would like to invite Professor Joacir de Oliveira. He will make the presentation of Professor Otávio Franco. Otávio Franco is professor at the where, Otávio? At Catholic University in Brasília, at PUC in Campo Grande. Okay. And Joacir will present Otávio, and I hope you are going to enjoy very much his talk. Thank you.
Ladies and gentlemen, good evening. It's a great pleasure to introduce Dr. Otávio Luiz Franco. Dr. Otávio Luiz Franco is a biologist from Federal University of Ceará. And he got his PhD here in this university, in University of Brasilia, in molecular biology in 2001. He did postdocs at Embrapa, also in Brazil, and at the University of Edinburgh in Scotland. His work focuses on the development of bioproducts and bioprocess with an emphasis on the control of pathogenic microorganisms. And he has worked on very important scientific missions in different countries, such as Australia, Israel, Portugal, Cuba, the Netherlands, and others. And a small one. Performed also sabbaticals in the area of infectious disease control at the University of Wisconsin, USA, and at University British Columbia in Canada. He is a 1A researcher and consultant for the National Council for Scientific and Technological Development, CNPq, here in Brazil. Agents of support, financial support in Brazil. His position is on the top. Dr. Franco is also a member of different scientific societies, such as Brazilian and the American Society for Biochemistry and Molecular Biology. Also a member of American Society of Microbiology and American Association for the Advancement of Science. His scientific production is impressive. He has published more than 500 papers, almost 40 book chapters, and 2 books, and at least 8 technological products. Dr. Franco, in innovation, he has more than 20 patents, tutored dozens of master's, PhDs, and undergrad students, and supervised many postdocs as well. So it's a great pleasure to receive Dr. Otávio, and the stage is yours, please. Thank you.
Thanks a lot. Meu prazer. Okay. Thanks a lot for everyone. I'm very happy to be here today. I will just give a small talk, and then we can discuss a little bit what I'm doing and what my groups are doing today. So I will just talk a little bit with the students, so in Portuguese. Primeiro, agradecer a todos vocês pela oportunidade de estar aqui e discutir com vocês. Outro dia eu era aluno da UNB, e eu me lembro exatamente a sensação de estar aí no lugar de vocês, só que vocês hoje têm um auditório mais bonito, mais legal. Eu era da época que era do fundo do subsolo, né, no subterrâneo, e é uma felicidade estar aqui hoje falando como palestrante, mas eu não me esqueci o quanto era bom estar sentado aí também. Tá? Just to translate, I'm just talking to the students. I'm very happy to be here, and that's in another day I was here like a student. It looks like maybe now it's 20 years ago. So, but I'm feel young like this. So today I will talk a little bit about in silico optimization. No, just here. Yes, you need to. Okay. Thanks. Let's try. Return. Nós não temos um pointer? No. Esse não tem pointer. Okay, no problem. So if I need to show something, I just put my finger and that's it, no problem. So today I will talk a little bit about in silico optimization of antimicrobial peptide and enable the development of bio-inspired products. So today I'm coordinating two groups, one in Campo Grande, that the name is Sinova Biotech, and the University Católica of Dom Bosco, and another one that's the oldest one. I have been coordinating this group in the last 20 years. The name is Centro de Análises Proteômicas e Bioquímicas. Okay. Tranquilo. And in both cases, I'm just loving peptides. So I'm trying all the time to transform these peptides in something that could be useful for the pharmaceutical companies, for the cosmetic companies, and for the agribusiness companies. So I'm trying to use a mixture of nature knowledge and also with algorithms and computational strategies. So I'm coordinating these two groups, and I'm also coordinating the Instituto Nacional de Ciência e Tecnologia, that the name is BioInspire, that is now in 22 countries. I have more or less 300 researchers involved in discovering new bio-inspired products. So this is the idea. I'm always glad to training students. For me, it's very important to show that science is very, very nice as a career. Today, I was in a big meeting in CAPS just discussing about the future of biotechnology. And this is one of the ideas that biotechnology could change a country like ours. So normally, as almost every person in this country, I start to use natural sources for finding peptides. So in the beginning, I just extracting plants, and in fact, I just starting plants. So I just starting with seeds and leaves and cerrado plants. So normally, most of people here are working all the time with cerrado plants. So I just discovered that some animals could be also interesting. So in this case, I'm using this one that is Stiella clava, that could be found in any part in the world, could be found here in Brazil and also in Canada. I spent some time in Cuba, so I'm working with mollusks, and I'm also working with coconut water, because I was interested in a legend of my grandma that just told me all the time, if you have some pain in the stomach, just need to drink some coconut water. And then this was very curious, because I'm just looking and see, almost everything that I can find here is water. But in fact, we just discovered a lot of peptides that could kill bacteria inside of the coconut water. And then we just studied the original sources. But the problem is, we can publish a lot. We can find a lot of peptides in everything that we can study. So I always told to my students, if you can bring a brick for me, we can try to find a peptide that kills bacteria, a brickinine or something. So it's not so difficult to find an antimicrobial peptide. If you spit in a bacteria, you kill it. But how could I transform this peptide in something useful? Because this now is the main problem that we have. For example, if you open an APD, that's a data bank of antimicrobial peptides, and you can find a lot of toxins over there. We can find maybe 5,000, 6,000 different peptides, but no one could reach the market. And I was just questioning to my team, what we are doing wrong? You know, I'm always telling to my students and always telling to all the people that we can find a new antimicrobial peptide or a new pharmaceutical, or create some anti-cancer peptide or something. But we have a lot of problems, like high complexity, low activity, a lot of high specific activity, because We are always trying to save our patients and not to kill them. A lot of problem with aggregation, difficult to purify, and many, many other problems. So what we need to do? So I will try to tell a history because I need to create a direction of a rationale for developing the best peptides. So we started with Stiella clava. This is a tunicate. And then a partner group from Netherlands just discovered this peptide that kills a lot of bacteria that the name is clavanin. Our group was not the first to discover the clavanin, but we are the first to elucidate the NMR structure. And what we observed, that when we have this peptide in water, we have a big coil. But when this peptide just binding to the surface of the membrane, then we have a helix. So we just observed that most of this peptide was just transforming from coil to a helix according to the environment. And then we just observed that these peptides have a lot of antibiofilm activity. So this we just used a microscopy and observed that in the case of Klebsiella pneumoniae, we just have a destruction of this biofilm. That's a lot of cells together. And in the case of staph, we just destroy most of the biofilms. And in this case, in the case of E. coli, we have more or less activity. Not so good, not so bad. And then we went to wound infections because I always told to my team that we can kill very easily the bacteria. We just can put some fire or burning, and that's it. But we cannot do this in an animal or in a patient. For example, what's the solution for the COVID? We can just burn the patients, and then the virus will be destructed. But this is not rational. So the same occurs with the bacteria. We can kill the bacteria very easily, but we cannot kill our patients. So we just start with a wound infection model, and then with 10 milligrams per kilo of the clavanin, we have almost 100% of control of our peptide, of the bacteria. And then we went to a sepsis model when we injected a lot of bacteria in the peritoneal of the mice, and then we observed that the clavanin was capable to control 80%, but 80% is not enough for a drug. Then we was just thinking, okay, we can use the original source, but we can just use this knowledge that comes from the nature and takes millions and millions of years to be here, and then we just can give an improvement. So we just used a strategy that we call an in-brain design. So in this case, we add a tag of 5 amino acids. We take a lot of peptides with immunomodulatory activities, and we make a huge alignment, and we find a pattern, because I really enjoy the patterns. And then when we add these patterns, we just add this tag with 5 amino acids and create a new clavanin, that the name is clavanin MO. But how this tag affect the structure? So we just observed that when you add this tag, the clavanin continues to be the same. So we have the same helix in the membrane structure, but we have in water environment the same helix, the same coil. So this is good because I don't want to change too much the structure. I want to improve the function. And then we tested this against the bacteria by the in vitro tests, and we observed that we improved a little bit the MICs, but not too much. So it was not so efficient. And then we just tested against fungi, and we observed that we have a real decrease in the MICs. So this showed me that our peptide was much more potent. And then we went to antibiofilm activity and observed that the clavanin MO was effective against Klebsiella, but was not so good against staph. And one thing, because normally I was always trying to understand how a peptide works. So in this case, we are just interested in a different mechanism of action. I was in Canada, and the people was just discovered that some peptides binding to this PPGPP, that is a tetraphosphate guanidine, And you observed that in this case was not the mechanism of action. So clavanin-MO did not bind into the PPP. And then it was once more to the in vivo tests and observed that clavanin-MO was capable to control by the same way of the original clavanin 100% of a bacteria in wound model, but in the sepsis model we have also the same. So we changed the activity. The activity improved a little bit outside of the animal, but inside was the same. So we changed the structure. We changed a little bit the sequence, but the sequence that was changing was not improved as I wish. And then we just crossed to the mastoparans, because one of my students was just telling me all the time, Octavio, please, let's work with the mastoparans. I say, no, I don't want to work with toxins. It's always difficult to sell to pharmaceutical companies. Okay, I can cure with venoms. But in fact, we just discovered that venoms could be very, very interesting to change. So we start with a mastoparan L, that is originally from wasps, with cationic and amphipathics properties, and it's very, very multifunctional. So they have a lot of activities. And then what we do, our first trial was to take the same tag and add to mastoparan. So we just add to this two mastoparans and then tested, and we observed that the activity was not improved as occurs in the clavanins. And then we just add this to brevinins and temporins that came from amphibians, and we test the potential of our tag. And once more, we just run a lot of NMR studies in order to understand if our tag was just changing the structure. Once more, we discovered that the tag did not change in the structure, and we construct this big panel of mastoparans, modified, and brevinins and temporins, and we observed that we have a good activity against most of bacteria. But then I was interested also in immune response. And in the case of the immune response, we have some changing when we add the tag to the mastoparan. So our interesting was to decrease the inflammation, because we was always involved in control the sepsis, and the sepsis is basically a storm of interleucins. So we are just interested in decrease the TNF alpha and improve a little bit the interleucin 6 and decrease the MCP. And we observed that mastoparan MO was capable to decrease the interleucin 6. And then once more, we went to the bacterial injection. So we just make a lot of in vivo models, because at this point we have maybe 50 different peptides, and we tested 10 of them against the bacteria in the model. No. And you can observe that when we improved the quantity of mastoparan MO, we have a very good control against this bacteria. But at this point, I was a little bit frustrated, because I can find one antimicrobial peptide, I can add a tag, and then I can test one single peptide. And then I just talked to a guy that the name is Kai Hilpert, that now is in Germany, and he just discovered a new technology that the name is spot technology. So he prepared these peptides and synthesize in a cellulose support. And in this case, it was much more cheaper. So normally, if there's anyone here that works with peptides, it's a little bit expensive. If I want to try 1,000 peptides, 2,000 peptides. If I want to try one, it's quite okay. But if I want to improve the number of the peptides, I need to have a more cheaper technology and a more fast technology. So in this case, we use it. I moved to Canada, and I was just connecting to Kai Hilpert to understand how this technology works. And the problem is, how could I create thousands and thousands of peptides? Because of course, if someone of you just try to isolate some peptides, it's not an easy task. And normally you have a simple sequence, and that's it. So we just created an algorithm that his name is Joker. So the Joker is a genetic algorithm that was used to create a higher variation of our peptide. So we just add to this algorithm the sequence of mastoparans, clavanins, and many others. So we have three kinds of residues that was evaluated by this Joker. The first one is the identity component. So in these huge alignments, all the time, I find that the same amino acid residue. So some are very well conserved, like lysines, cysteines, and other. So we have the second one, that is the ambiguous component. So I can change the amino acids a little bit, and when I change, I keep the classification, but I have a different amino acid with a different side chain. And then I have the wild card. What is the wild card? The wild card is like a joker. When you play cards, we have a joker, and then we add a different one. So we try to find in nature all the possibilities, and one that never was put in this position, we just added. So we created something really new from what nature offered to me. And then we add this in a scaffold of a peptide. So we just choose the best helix because I was interested to add this tag to these patterns in this helix. And then we just run like a sliding window, putting the different patterns in the beginning, in the middle, and at the end of the peptide. And then we created 300 derivated peptides. So was our first experiment. And then we have a success rate of 65%. That was quite okay. We have maybe 200 peptides with a good activity and low hemolytic activity. And to choose our leading candidates, we just modeling all of them and testing all of them. Okay. So in the case of the mastoparans, we created a lot of variations. So we have the R1, R2, R3, because since we have so many peptides, we are not so creative for names. Sometimes we will find something like PADBC1, T1 that my student like it, but I'm not. So it's normally we have a strange name. R1 and R2 was quite okay. And then we just explore the activity of this peptide. So the next step is we discover the peptide, we just check in a first trial, and then we go for a deeper study. So in this case, we just run some fluorescence microscopy and discover that R1 and R2 was very potent against Pseudomonas biofilm. So once more, we went to an in vivo test. In this case, was a peritoneal infection, and observed that mastoparans R1 and R4 are very potent. And then we just run some structural analysis showing that once more, I have a coil that was transformed in a helix. So normally I cannot change too much the structure. It's not so nice to see amazing structures because normally this kind of peptides are very helical. But we can just transform a toxin in something very potent with an activity that could be used for pharmaceutical companies. Another question is how the salt affects our peptides, because normally we have a problem with salt. When we improved the salt concentration, we lost the activity. And remember that one thing that we want is to have these peptides active inside of an organism. So normally I just tell to my students, remember that we need to inject or give orally or anything. And not 100%, but 99%, it needs to be potent and active. So in this case, we observe that salt, in the case of mastoparanel, it keeps the activity. It's good because you can understand a little bit why mastoparanel was so potent and have so slow specificity. But when we change, we decrease in 4 times the MIC, and R4 we decrease in 2 times the MIC. So when we have the salt, we did something in our peptide that transformed this peptide in a more stable peptide that could act in the presence of the salt. And then we just compare, for example, in the case of R1, when you have in clinical isolates, when you have 0 salt, we have an 8 and a 16. And we have, when we add the salt, we improve the activity to 2. So we need less peptide to kill more bacteria. So in this case, we just create an alternative for transform peptides, not only more potent peptides, but in peptides resistance to salt, and that could probably act better in vivo models. Once more, we just try to understand this relation. So we just run some CD analysis, and you observe that the salt do not affect too much the helix, even in environments of in this case, SDS, showing that we have 45% of a helix for R1 and 47 for R4. And then we run once more the NMRs, and you observe that we have a helix. This is in a membrane environment. We have a helix between the leucine 3 and the isoleucine 13 for R1, and the leucine 3 and isoleucine 14 for R4. So this just suggests me that we have certain stability when you change and create this new peptide that comes from osteoporosis. And then we just run some SPR. So we just run some surface plasmon resonance in order to see how these peptides binding to the lipids. And you observe that in the case of mastoperoneal, that is the original one, we have higher affinity against PCE and PG, and also POPC. That was the models of membranes that we have. And when you make all these modifications and change to R1 and R4, the association was lower. What this suggests me? For this reason, this mastoperoneal is so potent against everything. So it kills everything because it's not so specific. But when you change a little bit and transform the L in the R1 and R4, then we have a decrease of potency, but we improved the selectivity against the lipids. And then you run some leakage assays, and you observe that, for sure, these mastoperones are not so potent as melitins. In the case of melitin, it's a positive control that we have because it causes a lot of leakage, and show this in different models. Mastoperon L, once more, shows low selectivity, and R1 and R4 are 10 times less in comparison to parental. So in this case, we just transformed a low selective peptide to a very or more selective peptide in comparison to our original one. Finally, we just take the structure and try to understand how this structure binding to the membranes and how the membranes was disrupted. So we just run some simulations of molecular docking and dynamics, and you observe that in both cases, the peptides just binding to the surface of the membrane and then go inside. Now we are just trying to better understand, because in this case, I have one peptide and a lot of lipids. But in fact, in real life, I have a lot of these peptides that was just binding to the membrane, organizing the structure, and then causing the disruption. But we are just trying to improve the system because we need a lot of computational power. And then we just run some atomic force microscopy, and this atomic force microscopy show me that the peptide was just binding to the surface of the membrane and just destroying the surface, causing a reduction in the height and also decreasing the size of the bacterial cell. So at this point, was just working with mastoperons, and then we decide to improve it, because I'm always think that we can just improve it and improve it and improve it. Mastoperons is just one peptide, but normally we have a very amazing biodiversity in Brazil. So I have the opportunity to work in Cerrado and also in Pantanal, because I spend part of my time here in Brasilia and part of my time in Campo Grande. This is just 300 kilometers of the middle of the Pantanal. So when I was very, very stressed, I take my truck, I take my dog and go in the middle of Pantanal. I spend 2 or 3 days just looking the animals and walking and see what is good and very good for my life. So the government of Mato Grosso do Sul is just always asking me, Octavio, we need to get more studies in the Pantanal. So in this case, we just opened a new project where we just take a lot of species in Pantanal and run a genome sequence. So in my idea, the concept is we need to create a big data bank of a lot of information about blue macaws, tuyuyus, crocodiles, snakes, and everything that I can catch. So we just using a lot of high-seq technology and also uses some sequencing using by mini ions. So we just construct a laboratory in the middle of Pantanal that we can sequence the DNA maybe with 500-600 kilometers of nowhere. And then we have genome information. And using this genome information, I and my group finding a lot of encrypted peptides inside of these genomes. And then after that, we just mining with bioinformatic tools and just perform some chemical tests and just run some in vitro tests. And in this case, I need to combine Pantanal with cows, because when I move part of my time to Campo Grande, I discovered that they have much more cows than humans. So in the beginning, I was just thinking, I will try to find new pharmacies for humans. But then when I arrived in Campo Grande and Mato Grosso do Sul, lots of cows, lots of chickens, only few humans. So I think, okay, we can help the cows. So we just start to find new antimicrobial peptides. And in this case, I use an example of a snake, a Crotalus. And what we do, we just sequence the genomes and the transcriptomes of one of our Crotalus. And then we just find some natural peptides. And then using these natural peptides, in this case was a catelicidin from a snake venom, we just created thousands of bio-inspired peptides. And after create the bio-inspired peptides, we just tested and find a leading peptide and just go to the test. So these are our cows. In the beginning, the people does not believe that it was possible to transform a peptide in something for cows, and the government does not accept it that I can buy cows, so I buy my own cows. Now I have 10 cows. This is Mimosa. She is very nice. I really enjoy Mimosa, but she was very sad because she get mastitis. So for this reason, bioinspiration is needed. So we take a lot of species, and then this is not a real number. Now we just created and test 2,500 different peptides. And here I am in the beach with the alligators. They are very nice. So agricultural production in Brazil, milk production is the third largest milk producer in the world. 2020, there was an increase in production by 1.5%. So the problem that we need to solve it here is udder inflammation, because normally staph and other things go inside and cause a deep infection. And we have an economic impact, a reduction in milk production, prevention in treatment costs, contaminated milk disposal in zoonotical and food poison. So normally it's a loss of 35 billion annually. And so there's a lot of government that was interested to solve this problem. So staph is the main problem. So it's a bacteria that you can find in you and in me and normally in cows. And then for these 2,500 peptides, we find 1, 2, 3, 4, 5 good peptides that was very, very potent against Staphylococcus. The number of Staphylococcus is because I received it from the farms. And then we just focus in this one that we named MastPep. So MastPep is a product that we are developing now that was very potent against Staph. So in the beginning, it was difficult because we just need to test in vivo. I always say that we can kill stuff, but we need to transform this in something useful. And in the beginning, cows are normally very big and mice is very small. So I don't need a lot of peptides to test in peptides. So we just created a model of mastitis in mice, inducing the lactation. And you observe that with mastpep, we just have a good activity after 96 hours. And then with this indicative, I buy the cows, Mimosa just came to my life, and then we just created an infection method by using 10 cows. So we just make an infection in each one of the udders, and then we have 3 days of treatment. And what we observed that after 5 days, we have a control of the infection by using a peptide originally discovered from a snake venom. So it works, it's possible to transform something that is not cool, not very useful for the citizens by using bioinformatics tools. And one interesting thing is this peptide did not affect the tissue microbiota, so it just look into staph. And why also I just started this project. In fact, I was just thinking that I can contribute to a better food for our country. I remember the first time that I was in the US, I spent some time in Madison, Wisconsin, and then I was invited for a very nice dinner. And the guy that just received it, the name is Richard Burgess. I just talked to him and say, I like strawberries. I really like fruits. And then he gave me a very nice dinner with very amazing fruits, a huge mango that looks like a jackap or something, strawberries, bananas. And then I asked him, where did you find these bananas, man? And the guy say, it comes from Brazil. And I say, no, it's not possible. In Brazil, I have ugly bananas, and you offer me a very nice bananas. And then I look in there a seal that is bananas from Brazil. And I say, why in Brazil we need to eat the worst things? Do not make sense. We just produce this stuff, And the same is with milk, with yogurt and strawberries and blah, blah, blah. So I just thinking, why you cannot use our own technology, our own bioinspiration and a lot of biodiversity in Pantanal, Amazon or something to bring better food for our country. So in this case, I'm just trying to find a peptide that could kill the bacteria and that could not contaminate our milk with residues or something. And this is the concept that you are selling now to Ouro Fino Saúde Animal. That is now is trying this peptide in 400 cows. And it seems that it will be a good partnership. So another thing is because I don't want any residue. And here is the peptide. Here is the milk with no peptide because I do not put the peptide inside. And then when I put the peptide with the milk, after 2, 3, 4, 5 hours, the peptide completely disappear and we don't have any residue. So we have for a snake venom, a new peptide that could control the staph in mastitis and is a very, very nice green technology. So now, another problem is to produce this in large scale. So we just cloned this peptide inside of a bacteria. But imagine that normally the peptide killed the bacteria. So how could I clone it inside? So it's a trick question. And then we created a new construction that is a combination of our antimicrobial peptide, the intein, and elastin. So when this peptide is produced, it's a kind of gel. And then we just extract the gel from the bacteria and then give to the cow. So we have the production in a very large protein, and then we just remove the peptide with a decrease of pH and decreasing of temperature. And then we can find this in the mass spectrometry. And now we are just trying to show to the industry that it's possible to produce this peptide in large concentrations. So this is, and how much time do I have? Okay, perfect. So, Okay, so no, talking, my five, more five, 3, 2. Okay. If she leave me talking, I will continue and continue. I love this stuff, you know. So plant peptides could also be improved. So in this case, we used a toxic plant from guava. So at this period, I was very interested in plant peptides, and then I just buying a lot of guava. So I was in the Extra in the Asa Norte, and then I try to buy, because now the Extra is not there anymore, but I buy 300, 400 kilos of guava because I need to extract the seeds of guava. So I bring a lot of guava. For each 100 kilos of seeds, I have 1 milligram of the peptide purified. So imagine how much tons of guava I bring to my group. So my group have a lot of guava juice, guava sweets, beef with guava, everything with guava. The laboratory smell like guava. And then we just tested this peptide as native one, and it's toxic to most of stuff. And then we just producing recombinant way. But this was a not very easy peptide. I have a coil with a mixture of helix, and then it was difficult to produce this in large scale. So our first idea was to cleave it, and then we just have this. I just try to cut the peptide according to the properties that I was interested in. So alpha helix propensity, net charge, hydrophobicity, and hydrophobic moment, and nothing happens. So I do not have a good mix when I break the peptide. And then we just apply our algorithm and have 15 variations of this guanine, and then we have a very potent peptide. And then we tested this peptide against a lot of bacteria once more. And then we just try to evaluate the mechanism of action. In this case, we use electron microscopy and observe that this peptide was capable to bind into the membrane and cause perforations in the surface of the bacteria. Moreover, we also observed that it can cause a depolarization of the membrane, but was not so potent for that as a melitine. And then finally, once more, NMR structure showing that we have a helix in the membrane environment and a coil in the water environment. And then we went to in vivo assays, and once more, we have some activity. So it's possible now to transform venoms from wasps in something useful, venom from snakes in something useful, plants, toxins in something useful. But bacterial toxins could be improved, so we just go into the edge of knowledge, and then we take a toxin that is named Cre10A that normally perforate the surface of the midgut of insect, and it have been used a lot because it's isolated from bacillus, and it's used in agribusiness as control of insect. And we just observed that we have helix that could be useful to destroy the membranes, and once more, apply the algorithms and create new sequences and run some molecular modeling in order to understand this helix. And then the best ones was evaluated by NMR, showing that once more, we have the same pattern. So we have always a coil in water and a helix in a membrane environment. So at the end, we normally have the same patterns in animals, in plants, in bacteria, in everything that I can find it. So here is the panel that showed that this peptide was also active against E. coli and staph. And finally, just to finish, another peptide that we isolated from bacteria from Italy, a Pyrobaculum something. So it's very charming because it comes from a volcano or something. It's nice. And then we just take the original one and applied the algorithm to create a motif. So I was interested to understand the flexibility. If I change the flexibility, maybe I could control the specificity. So we just observed in the last 20 years that when you have more flexibility, we have lower specificity. So we just tried to prove this concept and then created this motif, NKP, and this NKP was inserted in this toxin from bacteria. And then we did this in several peptides, R3. In this case, this is a strange name, PADBS1, R2, R3, R3, R7. And then we run some molecular dynamics. And then we observe that this is true. We have a good activity when you add this NKP. And then we run some atomic force microscopy and observe that the peptide just destroy all the bacteria that we just challenge. And then we run some hemolytical and anti-cancer activity. And what we observed, that R7, that was the most flexible, was more active against cancer cells, human cells, and bacterial cells, suggesting us that when you improve the flexibility, we lost some specificity. Once more, we was to functional size and then run some activity against biofilms, and then all the set of data that you have been showing with in vivo activity, structural analysis, and other things. One thing that we observed in extra for this is because this peptide shows also the capability to fibroblast recruitment. So it's possible that this peptide could be used like helping in cicatrization. That was very nice for testing new antimicrobial peptides that could be active in the wound infections, because in this case, I have a combo that could kill the bacteria and close the skin and help in cicatrization. And now, what's the next? Because I always trying with my team, what's the next step? So we are just using and creating a new platform of deep learning or artificial intelligence to improve our knowledge against our knowledge about the antimicrobial peptide. So we are using an IMP prediction algorithm that we are using BERT, and then we have a reinforcement learning, and then we have an Xvii that will help us to create. So we have an Xvii that is explainable artificial intelligence. When we make an agreement with the guys that created the ChatGPT, so they offer us the scripts and the algorithms that we can change and add our parameters, and we are using Lime and Shape. And what this explains to us, for example, now we are just trying to understand what the impact of each of amino acids in the function of our peptide. So in this case, for example, aspartic, this is the first, we have a big blue line. So blue is bad. And in this case, red is good. So blue is terrible in most of peptides. When they have this aspartic, we can't do anything. But when you have lysine, that's our cationic amino acid, that is the red, the second line, this is good. So this is just helping me to explain what I'm doing with all of these peptides and designing better and better peptides by using the original sources. So it's just the same. So we have the worst and the best. So the concept is we have a prediction model that we just started with a natural source. And then we have now a big data bank, because normally we just publish the positive ones, but no ones want to publish the no MPs. Imagine I show you all the success that I have with 5, 6, 7 peptides. But at the end, I try with my team almost 5,000 peptides, and I have 4,000 peptides that are not useful for anything. So now I have a very nice data bank of something that I don't need to do, that is no MPs. And then we just use the Xvii and design model, new MPs, try, test, offer this information to the prediction model, and just running and let the system just learning and learning and learning. So now we're just using this strategy. We started with mastoparants, and it started also with maspep that came from a snake venom, and now we have an extra 500 peptides with different sequences. And the answer, it works I have no idea. Let's wait for the next talk, and then we discover what happens when I come in next year to talk to you guys. So I want to thanks to all of you. Thanks very much, Mariana, for the opportunity to talk. I'm really sorry to be running all the time and try to be here in the right moment. And I always try to help all the people that help me. So the people from the US, from Australia, from Lisbon, from India, from a lot of stuff, a lot of parts of the world. The people just believe in my crazy ideas and help me a lot, so that's nice. Also to financial support, CNPq, FAPDEF, FAPEMIG, Fundect, CAPS, UCB, and of course to all my students. So now my team is huge. I have now 120 students working with me, and it's a lot, but I really hope that they are working until now when I'm just talking and have fun with you guys. So thanks a lot. And this is my email, this is my Insta, and this is my TikTok. I do not make any dances, but I show a lot of signs. So if you want to follow, I will be there. Okay? Thanks for your patience.
Dr. Otávio Franco, thank you very much for your amazing presentation. Very great talk. We have time to a couple of questions. And se estudantes quiserem perguntar em português também, quem tem, às vezes está tímido até para falar inglês, como eu, então fiquem à vontade e podem perguntar em português também.
Otávio, congratulations for the great talk and great results. Impressive. I would like to ask two questions. One, you focus on the potency of the peptides. So you have very potent, very low MIC peptides. But how do they survive the proteolytic system on the host?
Works in a nanomolar. Oh, okay. In a nanomolar. So I'm trying to find more potent peptides. But then we have some potent peptides that are doing nothing, because when we injected, we lost 80%, 70%, and then we need to improve the doses to avoid the effectiveness of the proteinase. So now we are just inverting the L amino acids for D amino acids, especially for lysines and for arginines, because normally they are attacked to proteinase. So we are just focusing now in make a mixture of L and D amino acids and see what happens. But this is true. Or we can use some nanoparticles to protect. That is another part of the job that I'm not showing today, but this is very important. So I always focus in potency, but probably potency is not the main task at the moment to reach the markets.
Great, thanks. Another quick question.
Oh, sure.
You mentioned one peptide that was effective in sepsis, reducing IL-6. Did you test anything in the cellular response in the innate immunity?
Yes, we just tested in macrophage to see what happens when we mix with interferon gamma and see the panel of the interleucins. And normally it just decreased the TNF-alpha and the nitric oxide and the MCP1. I'm not sure if MCP1, but I believe it's so. So when I show this, probably it's not only for kill the bacteria. Kill the bacteria is the first step, but probably it's just modulating the immune response. I think that is also a key question for the antimicrobial peptides.
Thank you.
Thank you.
Another question? No? No, only one, two. Another, no.
In the tree.
Okay.
Okay.
No. Please, use the microphone.
Okay, I listen to you. So I was wondering, how are you going to look at how we're going to look, of course, at the differences between specificity, mammalian cells, human cells, and
Okay.
the peptides. That was the peptides that's going for the bacteria. That was what I was most interested in. Is how do you study this? That
Okay.
they're not killing the wrong cells.
Yes, this is also important because I don't want to kill my patient. So normally we have a lot of proportions between POPC and POPG, and also with the addiction of cholesterol. So we can mimetic some models to test, especially in ISPR activity. So we just create some chips with different compositions of the membrane and try to simulate what happens in a bacteria and also in a mammalian one. And then we go to in vitro analysis. So we have a huge number of mammalian cells, and then we test against leukocytes, macrophages, and other things. Most of them, and I think that is the most difficult for the toxins, is killing everything. So I always remember that I was working in Edinburgh, and then I was just creating a peptide that is destructing Trypanosoma brucei. And then I gave to the technician. The technician just performed the in vivo assay. And the other day, the guy say, "Congratulations, Octavio. It kills everything. It kills the mice. It kills the protozoa, and almost kills me." So in this case, we are always trying to design the peptides by using a different composition of lipids. The main difference that we have between bacteria and mammalian is because in the bacteria we have a very acidic surface. So normally the peptides have a cationic amino acid that try to select the acidic ones, but sometimes it's basically impossible. The peptide just destroy everything, and then we're just learning about this peptide. But the question is nice. Okay, thank you.
More questions? No?
Thank you for the tremendous talk. I have a very minor question. You were telling that it does not affect the tissue microbiota. So can you explain like how it affects the pathogenic microorganism, and it does not affect the normal flora or tissue microbiota?
Okay, this is also a very amazing question because normally the people are just finding antibiotics. In my case, I'm trying to find some selective biotics because when you kill the bacteria of intestine and stomach, it causes a harmful effect. But my answer is I have no idea. I think that I'm a lucky guy because sometimes we find something very specific, and I'm really trying to understand what happens, but I don't know. Sometimes we can find something that show different specificity because we have some activity against the cell wall, and then we can easily just transform to this part or this part. But normally I have no idea. I'm sorry.
Thank
I
you.
hope to answer this in 5 years.
Actually, this question suddenly came in my mind. I also don't know,
Uh-huh.
but this is a very critical question, how it
Yes,
does
for
not
sure.
affect the tissue microbiota or other microbial flora in
Exactly.
the humans.
I think that the guy that solved this problem will publish a lot in Nature and get it rich because it's
Okay, thank you. No.
I think the last question, Maria Elena.
Thank you for the nice talk. With this huge production of peptides, have you already transferred someone for the technology for the use? Because I think it's the main objective. no?
Yes, for sure. This is an important question because normally we are just focusing transfer this technology. Our first product was with a cosmetic company that the name is One Skin. We just produced a new anti-aging peptide, and it works because I have 110 years, so but it works. So we just transfer the technology, and now it's selling in Europe, in US, not here in Brazil. And now we just signed a contract with Orofino for this mass pep for control mastitis. But our problem now is to help them to produce in large scale. This is a huge challenge, because when you can reach to maybe 10 milligrams per liter, that is not too much. We don't could reach. So they expected that we offer them 1 gram per liter, and we are just working very hard on this. In the case of mastitis, it's a real problem, because we need to lower the prices and offer them goods and cheap products. In the case of, for example, a pharmaceutical company that you have more aggregated value, then we don't need to go to a heterologous system production. We just synthesize, and that's it. Like in cosmetics. In this case of cosmetics, one small flask is almost $200, and they sell a lot, so we just expend $20, and so in this case, we don't need to produce in heterologous system. So it depends of the market, but this is a challenge. Offer some product that have some aggregated value and could be sold in a very nice price.
Thank you.
I will give the. Can be.
Yeah, sure.
What about the potential for development of microbial resistance to your peptides? Because obviously that's a problem with current
Yes.
antibiotics.
Yes, for sure.
And if you're going to use it in animals at large scale.
Yes. In the maybe 5 years ago, I just tell to everyone, no worries, my peptides do not develop resistance. And now we are just publishing with Wagner now a very nice paper showing that after 40 rounds, just challenging the bacteria with our peptides, we have huge resistance. So it's the same problem that we have in the original antibiotics, like kanamycin or something. We just need to work with a very low concentration, and we need to change or mixture the antibiotics to avoid the resistance. But I think that at the end, I will need to keep going and find new antimicrobial peptide. When I sell the first, I just need to offer them the peptide 2.0, 3.0, because the resistance will appear. And this is a real problem in the animal market. So we just need to organize to keep the concentration very low. If not, we have a real problem in 10 years or 20 years.
Dr. Otávio, thank you once more. And I give the Mariana, please. Thank you very much.
Otávio. Otávio, thank you very much for an amazing experience. And now I would like to invite all of you to our welcome cocktail. Please join us outside. Foi, né? Foi?